Hi everyone, this is going to be the first lesson on the topic of organisation for GCSE biology students. In this topic, we're going to be expanding upon some of the ideas that we encountered in the previous topic on cell biology and looking at the ways in which cells are organised into tissues, organs and organ systems, and taking a look at specific types of organ systems, organs and tissues in different types of organisms, plants, animals and so on. In this lesson, we're going to outline the fundamental ideas in organisation in biology, and we're going to start by recapping some of the important ideas from that previous topic on cell biology. So in this lesson, we're going to be looking specifically at what constitutes a tissue, an organ, and an organ system. How are cells organised in biology? And more importantly, perhaps, why are they organised in this way? So let's take a look at as I say, some of the fundamental ideas from the previous lesson. Now, as always, I'm joined by a number of students via the Zoom application. Many of you have asked a number of important and relevant and insightful questions concerning the topic for today's lesson. I will be addressing those, as always, in the Q&A discussion session that will follow the recorded segment of this lesson. So those of you in attendance with me via Zoom, please hold fire on those questions. And if any other questions occur to you during the course of this lesson, then you can type those into the Q&A box in your Zoom client, and you can vote on those questions as the lesson progresses. And then at the end of the recorded segment of this lesson, I will address those questions in order. Now, some of those questions I'm going to be addressing throughout the recorded segment of this lesson, but as always, if you're watching this as a recording, and you have questions about the content of this lesson, you can always send me a private message via the this lesson's page on the website. So you can simply log in, go to this lesson's page on the GCSE biology website, and you can send me those questions as private messages via the student notes function, in which case I'll respond to your questions within 24 hours, typically via private message. If you'd prefer, you can type your questions into the live chat on this lesson's page, and perhaps start a discussion with other students on that page where I will respond. Alternatively, you can always email me and I'll be happy to answer any of your questions that way. If you're watching this as a recording elsewhere, such as on YouTube, then by all means type your questions into the comments and I will do my best to get back to you there as well with some answers. So before we go any further, then let's take a look at the learning objectives for today's lesson. So once you've watched this lesson video, if you're watching it as a recording, and if you're watching it on my website, once you've completed the various questions that will pop up at intervals throughout this video when the video pauses, together with the other learning activities on this lesson's page on the website, such as the end of lesson quiz and so on, you should be able to do the following things. So we first need to start by defining our terms. What do we mean by the terms tissue, organ, and organ system? Now, we're also going to look at something that we encountered in a previous lesson in that last topic on cell biology, the idea that single-celled organisms and single cells in general are limited in their size and their growth by the surface area to volume ratio that they have and the necessity for them to obtain the necessary substances to support their metabolism, things like oxygen, for example, by diffusion through their cell membranes. The idea that surface area to volume ratio will restrict the maximum size that a cell can grow to, because beyond that size, its surface area to volume ratio is no longer feasible for diffusion to satisfy the needs for transport of substances. So we will then go on to look at the way in which cells are organized into the most important types of animal tissues and also plant tissues. We're going to be looking at the main types of animal tissue, the specific specialized cells that make up those tissues, and relating the structure of those tissues to their functions. We're going to do the same for the most important types of plant tissues as well. And then we're going to go on to look at the structures of some human organs and also the structure of a leaf as an example of a plant organ. And the whole idea behind this is to lay the groundwork for the remainder of this topic, where we're going to be looking at various organ systems and the way in which they work. Now, in particular, we're going to restrict ourselves to looking at a couple of organ systems in humans, so the digestive system, the circulatory system, and the respiratory system, and looking at the ways in which cells and tissues and organs in those systems work, relating their structures to their functions. but we're also going to be looking at exchange and transport systems in plants as well. So obviously the groundwork for all of that is going to be laid in this lesson, where we look at these fundamental concepts of the ideas of tissues, organs, organ systems, and the need for this level of organization in biology. So as you can tell, there's an awful lot to get through, so let's make a start then. So let's quickly recap some basic concepts now. So the idea of a cell is something that we covered in a previous lesson, where we defined the cell as the smallest living unit of matter. And this begs the question, how does a biologist distinguish from the living and the non-living material universe? What constitutes a living thing? Well, by almost universal agreement, biologists have chosen to define living things as those which can perform 7 characteristic functions. Now, these include the following: respiration, movement, sensitivity, nutrition, excretion, growth, reproduction. Now, all of these are characteristic of living things. That is to say that any living thing of any size or scale will perform these 7 characteristic functions. Now, this includes unicellular organisms, individual, free-living, single-celled organisms. They are defined as being living because they perform all 7 of these functions. Now, according to some biologists, homeostasis, the maintenance of a constant internal environment despite fluctuations in the external environment, this is also a characteristic function of living things. But for now, we'll restrict ourselves to these things. So a cell is defined as the smallest piece of matter, the smallest material object that will perform all 7 of these characteristic functions of life. Now, in more complex multicellular organisms, cells are organized, specialized cells are organized into tissues, organs, and organ systems in order to maximize the efficiency with which these processes occur. So we encountered the idea that there are such things as free-living unicellular organisms, which is each made up of a single cell. So things like the amoeba, for example, or a bacterium. These are single-celled organisms where the living thing is a single cell. But in multicellular organisms, we have specialization of cells. Now again, the idea of specialization is something that we encountered in that previous topic on cell biology. So if you need to recap what we mean by the term specialization, I would suggest going back to the 2nd lesson in that series, and also taking a look at the 5th lesson, where we talked about the process by which cells become specialized, which we call differentiation. Now, in multicellular organisms, specialized cells are organized into tissues, and we used the analogy that just as a wall is made up of smaller units called bricks, animals and plants, multicellular animals and plants, and multicellular organisms in general are made up of smaller living units that we call cells. So here we can see a section of a leaf viewed under an optical microscope, and we can see that it is indeed made up of smaller building blocks, just as the brick wall is made up of individual bricks. Now, we talked in the previous topic, where we looked at transport of substances across membranes, about the fact that cells are limited in their size by this principle of surface area to volume ratio. Now, a single-celled organism, such as an amoeba, has to obtain all of its nutrients, all of its oxygen, and also eliminate all of its waste products across its cell surface membrane. And so, single-celled organisms typically have to have a large surface area to volume ratio. And this means that dissolved substances don't have very far to travel to get into or out of the organism, because in these cases, diffusion and other processes across the membrane are entirely responsible for supporting the metabolism of that organism. So the word metabolism means the sum of all chemical reactions happening inside an organism that support its life. So in a single-celled organism, all of the chemical reactions and processes that occur inside that organism that sustain its life have to be powered, have to be supported by the transport of substances into and out of the cell, and this all has to occur across the cell surface membrane. And so the bigger the volume of the cell gets, the more of those processes have to be supported. But as we saw in a previous topic, in the previous topic where we looked at the transport of substances, as the size of a cell increases, its surface area does not increase in proportion to its volume. And so there comes a point where the surface area of the organism is no longer sufficient to support the metabolic activities occurring within its volume. So in cases where we're talking about single-celled organisms, the process of diffusion, typically for a single-celled organism, is adequate to supply the organism with all of the materials that it needs, such as oxygen and nutrients, and also the removal of waste products such as carbon dioxide. And again, this is because the distance over which the materials have to diffuse is relatively short. So let's take a look again at the principle of surface area to volume ratio, just to illustrate this, because this is essentially the reason why you can't have single-celled organisms such as amoeba grow to the size of, say, blue whales, because eventually the cell will reach a size where its surface area is not sufficiently large to service the needs of its volume. So let's take a look at a cube. Let's imagine we have a cube with sides of length 1 millimeter. And so if we calculate its surface area and its volume and compare those to each other as a ratio, we see that because the cube has 6 faces and each of those 6 faces has an area of 1 square millimeter, the total surface area of this cube is 6 millimeters squared. Its volume is simply going to be its height multiplied by its width multiplied by its depth, and that's going to give us 1 cubic millimeter. And so we see that it has a surface area to volume ratio of 6:1. So let's imagine now that this cuboidal cell, this cube which we're using to represent a single cell, doubles in size in terms of the length of its sides, going from 1 millimeter to 2 millimeters. Let's see what that does to its surface area to volume ratio. Well, when we calculate the surface area and the volume of this cube, we find that its surface area is 24 square millimeters and its volume has now gone up to 8 cubic millimeters. Now, if we represent this as the lowest whole number ratio, we see that the surface area to volume ratio is now 3:1. So, by doubling the length of the sides of the cube, we have halved the surface area to volume ratio. If we then increase the size of the cube further to edges of length 3 millimeters, we can see now that this cube has a total surface area of 54 square millimeters and a total volume of 27 cubic millimeters. Now this equates to a surface area to volume ratio of just 2:1. So as we can see here, as the size of the cube increases, as the length of the sides of the cube increase, we can then go on to calculate the surface areas and the volumes of these cubes, and then have a look at how the surface area to volume ratio changes in relation to the sides of the length of the sides of the cube. And what we see is that as the length of the sides of a structure increase, if we're talking about a cube, the surface area and the volume will both increase exponentially, but the volume will increase at a greater rate than the surface area does. And this makes sense because to calculate a volume, you're multiplying 3 dimensions. You're raising something to the power of 3. Whereas if you're trying to calculate an area, you're simply raising something to the power of 2. And so we see that as the size of the object increases, its surface area and volume both increase exponentially, but the volume increases at a faster rate than the surface area does. So if we look at this then, we see that the surface area to volume ratio, if we plot the surface area to volume ratio and how that changes with the cube's length of edge, we see that the surface area to volume ratio decreases exponentially as the cube's length of edge increases. So what this means is that as the size of a structure or an organism increases, the surface area does not increase in proportion to its volume. So this means that once a single cell reaches a particular size, there comes a point where its surface area is not sufficient in terms of diffusion, active transport, facilitated diffusion, in terms of transport of substances across that surface area. It's no longer sufficiently large to for those processes of transport to be sufficient to support the internal chemical reactions, the metabolism happening inside the volume of that cell. So multicellular organisms have to rely upon processes other than simple diffusion in order to maintain the life of that organism. So the process of cells dividing is triggered essentially, at least in part, by the cell reaching a size where it has to divide in order to increase its surface area to volume ratio so that the daughter cells can continue to live. But also the development of multicellularity, the evolution of multicellular organisms, is itself partly a consequence of the increased efficiency of multicellular structures in terms of their surface area to volume ratio. So in larger multicellular organisms, diffusion becomes less efficient because the surface area does not increase in line with the size. So multicellular organisms have evolved various transport systems to carry dissolved substances that they need to the cells where they're needed. So the exchange surfaces in these multicellular organisms themselves are also highly modified to maximize the surface area to volume ratio. Now, this entire topic is going to be looking at various transport systems, mass transport systems, and exchange systems, such as in the lungs of a mammal. And we're going to be looking at the ways in which the tissues and organs within those systems are adapted for those functions. But all of this goes back to the significance of surface area to volume ratios. So in multicellular organisms, the cells and tissues do not all perform the same function. There is a division of labor. So an analogy that you can use to describe this is that at school you're taught different subjects by different teachers who are specialized in one or perhaps two subjects which they know very well. And this ensures that you're effectively and efficiently educated to a high standard in those subjects. Now, there's an old saying which I'm sure you've come across, the idea that a jack of all trades is a master of none. So to make the organism even more efficient at survival, at achieving the 7 characteristics of living things, the tissues are further organized into organs and ultimately into body systems. So let's take a look now at one final look at how this works in terms of surface area to volume ratio, how the jump from a single cell structure to a multicellular structure improves surface area to volume ratio. So consider the following tubular and cubic structures. These both have a total volume of 8 cubic millimeters. So let's look at the cube again, which has sides of length 2 millimeters. Again, as we saw before, the surface area to volume ratio of this cube is 3:1. Now let's, instead of this structure, which has a total volume of 8 cubic millimeters, let's instead imagine that we take 8 single cells, single cubes, each of which have sides of 1 millimeter, and stack them into one long tubular structure like this. So if we look at what this does to the surface area to volume ratio, we now see that the total surface area of this structure is 34 square millimeters. We have the ends, so 1 square millimeter here, 1 square millimeter at that end, and then we have 8 times 4 for the sides of this structure here. So this gives us a total surface area of 34 square millimeters. We still have a total volume of 8. So we can see here that we've gone from a surface area to volume ratio of 3:1 for a cuboidal structure with this volume to a surface area to volume ratio of 4.25:1 for this lengthened, elongated tubular structure. So the elongated structure has a greater surface area to volume ratio than a cube-shaped structure, and we'll see the importance of this later on where we look at the digestive system and the importance of the villi and microvilli. So cells within the body of a multicellular organism are organized as follows. So you have the fundamental building block of life itself, the smallest living unit of matter, the cell. Cells then differentiate into specialized cells, as we saw in that previous lesson on differentiation and specialization in the last topic. So cells can specialize to perform specific functions. They develop specific structural features through the process of differentiation that allow them to perform specific functions. But then, if you take specialized cells of a similar type and group them together, you form a tissue, and the tissue will then carry out one or more definite functions within that multicellular organism. Different tissues can then be organized into functional structures called organs, and those organs can then carry out one or more definite functions within that multicellular organism. But then different tissues and organs can all work together in a specific way to form a system, an organ system that carries out one or more definite larger scale functions within that organism. And finally, all of these different tissues, organs and organ systems work together to form an organism, a multicellular living thing that carries out all 7 of the characteristic functions of life. So, tissues, organs, and organ systems are levels of organization which allow for the more efficient carrying out of those 7 characteristic functions of life. So let's now take a look at some specialized animal tissues to exemplify what we've been talking about. So there are about 20 different types of specialized animal cells and many subtypes of each of these, and they all contain the following basic structures that we saw in that previous topic on cell biology. There's a nucleus, there's a cytoplasm, there's a cell membrane, there's mitochondria, ribosomes, and so on. So all of these structures are common to all of these specialized animal cells. They're necessary for the functions of all of those cells. Now, I say all, there are some exceptions. So for example, red blood cells will lack a nucleus, but in general, almost all specialized animal cells will contain these structures. But their overall structure is going to be different depending on the specific functions of that particular specialized type of cell. So, when we organize specialized cells of a particular type all together in one place, we've formed a tissue, and there are 6 principal kinds of animal tissues. So if we look in humans, for example, we will find epithelial tissue, connective tissue, skeletal tissue, blood tissue, muscle tissue, and nervous tissue. These can be thought of as the 6 main types of tissues. And again, a tissue is a group of similar specialized cells that work together to perform a particular set of functions. So these 6 principal kinds of animal tissues, with subtle differences, are found in many different kinds of animal. We're going to be looking at them in humans in particular, but they're found in all kinds of different animals. So epithelial tissue is perhaps the simplest type of animal tissue. It's made up of a sheet of cells that fit closely together, kind of like the slabs in a pavement. So in fact, one type of epithelium is actually referred to as pavement epithelium. Pavement epithelium is found covering the body surface and various organs. Now, pavement epithelium also forms the lining of some of the tubes in the body, such as the mouth and also the air sacs in the lungs, and it also forms the innermost layer of cells in the walls of arteries and veins as well. So here we can see a micrograph with stained epithelial cells from a cheek swab. Now, there are many different kinds of epithelium, not just pavement epithelium. All epithelial cells surround the edges of organs or the inner linings of tubes and cavities in the body, but there are several different variants, each of which performs one or more slightly different functions. So we have cuboidal epithelium, we have squamous or pavement epithelium, we have columnar epithelium, we have stratified epithelium, we have glandular epithelium, and we also have ciliated columnar epithelium. These are all different kinds of epithelial tissues that we're going to be encountering all the way through this topic when we look at the structures of various tissues and organs in the various body systems in humans. For example, we're going to encounter glandular epithelium where we look at the digestive system. We're also going to encounter ciliated columnar epithelium, for example, when we look at the respiratory system and the type of epithelium that lines the airways of the respiratory system, and so on. Now, connective tissue is literally just tissue that connects other tissues together. So the types of connective tissue are things like ligaments, tendons, meninges, which are the fibrous layers of tissue surrounding the brain, collagen, bone marrow, fat cells, or adipose tissue. These are all technically different types of connective tissue. So if we, those of you who are perhaps vegetarians or vegans, you might want to look away now. So this picture is of a beef steak, and what we see here is the connective tissue in this white color here. Under that we see the more reddish color of the muscle tissue. So skeletal tissue supports and protects the body's delicate organs and also allows for movement because of the attachment of muscles to the ends of the bones of the skeleton. So very often we refer to the muscles and the skeleton and also various connective tissues that connect them together as the musculoskeletal system. So here we can see a thin section of bone when observed under an optical microscope at a magnification of about 1,000 times. Here we can see the bone cells, and here we can see deposits of calcium phosphate, which is the mineral which gives bones their density and hardness. Blood is also characterized as a tissue because, again, it is a collection of similar specialized cells in one place. Now, the fact that it's a liquid doesn't change the fact that it actually does conform to the definition of a tissue that we gave earlier. So here we can see how a blood smear appears when observed at a magnification of about 1,000 times under a microscope. So blood is in fact a complex tissue containing a number of different types of specialized cells suspended in a fluid matrix. So the fluid that we find the cells suspended in is referred to as the blood plasma. Here we can see the platelets, which are fragments of cells involved in blood clotting. These cells here are white blood cells, and of course the rest of them are the biconcave disc-shaped red blood cells that you see here. Now, red blood cells, which are technically referred to as erythrocytes, these contain a respiratory pigment called hemoglobin. This carries oxygen around the body and delivers it to the tissues. Mammalian red blood cells don't have a nucleus when they mature, and they have this biconcave disc shape that you see here. Now, the biconcave disc shape of the red blood cells and also the fact that they don't contain a nucleus, together these features allow them to maximize their surface area to volume ratio, which in turn allows them to carry the maximum amount of oxygen from the lungs to the tissues where that oxygen is needed. Now, the other feature that these have is that they're highly flexible. They're able to squeeze and fold and pass through the narrowest capillaries, and this in turn allows them to deliver oxygen to the tissues more efficiently. Now, as you can see in this photo, the diameter of red blood cells is almost the same as the diameter of the capillaries through which they pass. Red blood cells typically pass through capillaries one cell at a time in single file. Now, this means that there's less distance and a larger surface area for gas exchange to occur between the walls of the red blood cell and the capillary wall, and this makes the process of diffusion of oxygen from the red blood cell outward through the wall of the capillary and into the surrounding tissues more efficient. So muscle tissue is made up of specialized muscle cells. Here we can see a micrograph of individual muscle cells or muscle fibers. You can see that they have this striped or striated appearance. So these types of cells, these skeletal muscle cells that you see here, are specialized for contraction, for getting shorter and generating a tension, a force, which can then be used to move the muscles and move the bones. So the individual muscle cells here, again, are specialized to perform that function. Their structure contains specific features which allow them to contract and generate forces. And this is important in the function of movement, again, one of the characteristic functions of living things. Now, the type of cells that make up heart muscle tissue are rather different to those making up skeletal muscle tissue. Heart muscle tissue doesn't ever get tired. It's constantly contracting and relaxing, and unlike skeletal tissue, it will never stop doing so. And so obviously it needs to be structurally quite different to skeletal muscle tissue. So cardiac muscle is the type of muscle tissue only found in the heart. Rather than having linear fibers, as you saw in the skeletal muscle tissues, it has branched fibers which contract rhythmically. And the branched arrangement allows for the whole heart to squeeze, to contract around the volume of blood inside it, to allow the heart to fulfill its function of pumping blood around the body. So if we zoom in on a section of heart muscle tissue, we will see that the cardiac muscle fibers have these branch points between them, as you see here, which are again quite different from the type of structure that you see in skeletal muscle tissue. If we were to look at cardiac muscle under a microscope, we might see something like this. So here, again, we're looking under an optical microscope at a magnification of around 1,000 times. And again, you can see here the muscle cells, the nuclei of those muscle cells, and you can also see how they're branched and linked to each other in a branched arrangement. Now, nerve tissue is made up of long, thin cells which transmit electrochemical impulses, nerve impulses around the body. And these nerve impulses can travel very long distances, such as between the brain and the muscles of the big toe. So here is a diagram of a type of nerve cell. So you can see here the cell bodies can have these many branching points through which they can contact and synapse with other nerve cells. And again, we're going to be looking at the nervous system in a later topic where we talk about homeostasis and the response to stimuli. This is another type of nerve cell here called an intermediate or relay neuron. So there are many different kinds of nerve cells or neurons. They have many different types of structures, but they all have in common the ability to pass these electrochemical impulses. If we were to look at some neurons under an electron microscope, we could actually see the branched nature of these cells quite clearly. So we've been taking a look at various types of specialized cells and also types of tissues that you find in animals, exemplified by human cells and tissues. Let's take a look at some organs. Now again, we're going to be revisiting many of these organs in later lessons, both in this topic and in subsequent topics. But let's just take an overview here just to see how cells are, or rather how tissues are organized into organs. So the stomach is an example of an organ. It mostly consists of epithelial and muscle tissues. Here is the location of the stomach in this model. And the tissues in the stomach wall produce both acid, hydrochloric acid, and digestive enzymes, in particular protease enzymes such as pepsin. And the whole organ has a muscular wall which contracts to churn the food into a liquid called chyme. The heart is another example of an organ. It mostly consists of a specialized type of muscle tissue called cardiac muscle tissue. And again, the structure of the heart is related to its function. The tissues in the heart wall contract rhythmically, and the whole organ pumps blood to the lungs and all the cells, the tissues and organs of the body. The brain is another example of an organ. It's composed mostly of nerve and blood tissues. So here we can see on the left a photograph of a brain, and on the right we can see a diagrammatic representation of this. So here we see the cerebrum. We can also see other parts of the brain. This is the part of the brain called the cerebellum, which is responsible for coordinating movement. Down here we can see the spinal cord where it meets the brain, and at the bottom of the brain we have a part of the brain referred to as the medulla oblongata. So the brain, the function of the brain is to control movement and other body functions. Now again, the structure and function of the various parts of the brain is somewhat beyond the scope of GCSE biology, but it is important to know that there are main functional areas of the brain. So here we can see some of those areas highlighted. So at the back of the brain we have an area of the brain on the surface called the occipital cortex, which is responsible for the assembling the data, the information coming in through the optic nerve from the eyes into our perception of the visual field. We also have areas of the brain involved in hearing and speech, such as Broca's area. We also have the sensory cortex, which is the part of the brain responsible for interpreting the signals received from sensory organs in the skin, for example. And we also have next to that an area of the brain referred to as the motor cortex, which is responsible for the sending of signals to the muscles to cause contraction and movement of the limbs. Here, these two halves of the brain are referred to as the left and right cerebral hemispheres, which make up the cerebrum. At the back, we have the cerebellum, and at the bottom, we have the spinal cord here. Now here again is the medulla oblongata. This is involved in regulating involuntary activities such as heartbeat and blood pressure, for example. Now this again seems quite complex, but this in fact is a huge simplification. All we're looking at here are some of the external structures. Within the brain, there are various internal structures, and even within these areas, there are subdivisions which deal with specific functions of sensation, of speech, of cognition or thinking, and so on. So we've looked at tissues, we've looked at organs. Let's now take a look at the idea of an organ system. So once again, an organ system is made up of specific organs and tissues which work together to perform a particular set of specific functions within that multicellular organism. So we have the digestive system, we have the muscular or musculoskeletal system, we have the circulatory system, we have the nervous system, and we have the reproductive system, but we also have other systems such as the respiratory system and the endocrine system. Now all of these are collections of tissues and specific organs which have specific jobs to perform within the body. So we've taken a look at animals. Let's now move on to look at plants. So in the exact same type of division of labour exists in multicellular plants. These are made up of thousands of specialised cells grouped into tissues, organs and organ systems. Now there are many different specialised types of plant cells, but we can broadly say that they all contain the following basic structures. They contain a nucleus, a cytoplasm, a cell membrane, mitochondria, ribosomes, and there are specific types of cells which, as we'll see, form photosynthetic tissues which also contain organelles referred to as chloroplasts, which are the site of cellular photosynthesis. So this is a diagram of a plant cell. Here we see the cell membrane, we see mitochondria, we see the cytoplasm, the nucleus, and we also see the ribosomes here. Now, as in animals, the overall structure of plant cells is specialized into particular types of specialized cells, and these specialized cells allow these multicellular plants to carry out one or more particular functions which relate to the characteristic functions of life much more efficiently than they could if you were dealing with single individual cells. So let's take a look at the main types of plant tissues now. So again, like animals, plants have a level of organization based upon cells being organized into tissues, tissues being organized into organs, and then organs being organized into organ systems. There are 6 principal types of plant tissues. Now again, this is a bit of a simplification, but for GCSE purposes, this is sufficient. So there is epidermis tissue. Now, this is the type of tissue which typically forms an outer protective layer. There are photosynthetic tissues, which are composed of cells which contain chloroplasts for photosynthesis. There are also vascular tissues. Now, the term vascular is used in both animals and plants to refer to, in animals, for example, to refer to the circulatory system, the vessels of the circulatory system, such as arteries, veins, arterioles, venules, and capillaries. So those are the vascular system in animals, and it is a transport system. But also in plants, you have a vascular system, vascular tissues, which are responsible for the transport of substances. And in plants, this includes the xylem tissue, which transports water and dissolved minerals from the roots to the parts of the plant that require it, in particular the leaves and the shoots. And you also have the phloem tissue, which is responsible for transporting the products of photosynthesis, sugars and also other assimilates, other substances, to the growing parts of the plant from the leaves. You also have meristem tissue, which is the tissue within which rapid growth and differentiation takes place. So meristem tissue is typically found in the growing regions of the plant, the tips of the shoots and the tips of the roots, as we saw in that previous lesson in the last topic on cellular differentiation. In plants, you also have tissue which bulks up the stem and the roots, called packing tissue. And finally, you have a type of tissue called strengthening tissue, in which you have cells with thickened cell walls to provide strength and support for the plant so that it doesn't fall over as it grows upwards. So let's now take a look at each of these tissues in turn and relate their structures to their functions. So the plant equivalent of the epithelial tissue that we find in animals is referred to as epidermis tissue. And again, this protects the structures that lie beneath it in the plant. So if we take a section through a leaf, such as being shown here, we take a leaf, we take a thin section through it and we view it under a microscope, we're going to see that the outer region, the outer layer of cells which protect the underlying regions of the tissue of the leaf, this layer is referred to as the epidermis. So in this micrograph here from an optical microscope, we can see the epidermis of the onion here. And you can see that the cells are linked together in a very similar way to the type of organization that you find in the epithelium of an animal. Now, the roots of plants are responsible for absorbing water and dissolved mineral ions from the soil. So here we can see that the roots have this fine covering of root hairs. So roots are often covered in thousands of fine extensions like this called root hairs, which are modified epidermal cells. Now again, the reason for this structure is to maximize the surface area of the root so that more efficient absorption of water and dissolved minerals can take place. And so for this reason, the epidermis of a root that you can see here on the right has these fine root hair cells which have specialized to produce these long projections, and this in turn maximizes the total surface area of the root for more efficient absorption of water and dissolved ions from the surrounding soil water. So, let's take a look now at photosynthetic tissue. So the two types of photosynthetic tissues that you typically find in the leaves are referred to as the palisade and spongy mesophyll tissues. So these are the layers within a leaf. So again, if we take a section through a leaf and view it under an optical microscope, we would see that we have two layers of cells here, which are photosynthetic. So here we see the palisade mesophyll, and here we can see the spongy mesophyll. Both of these layers of cells contain chloroplasts, a large concentration of chloroplasts within them to maximize the efficiency of photosynthesis, to capture the maximum amount of sunlight energy passing through the leaf. Now, in fact, the epidermis layer and the upper and lower epidermis layers and also the guard cells themselves also contain relatively smaller numbers of chloroplasts, but it's really these two layers of photosynthetic tissue where you find the largest amount of chloroplasts per cell. Now, the leaf is actually an organ. It is a collection of tissues which are specialized to perform a particular function, specifically the function of photosynthesis. The leaf is the photosynthesizing organ of the plant, and for this reason you have photosynthetic tissue, but you also have transport tissue, vascular tissue, xylem and phloem. The xylem to carry water and the dissolved minerals necessary for these cells in the leaf to photosynthesize. So the xylem tissue delivers that water from the roots to the leaves, but you also have phloem tissue in these vascular bundles which make up the veins of the leaf, and the phloem tissue is responsible for transporting the sugars which are being made by the photosynthetic tissues back to the growing regions of the plant to the tips of the shoots and the tips of the roots. So again, you have spongy mesophyll, you have a lower epidermis, and you have these guard cells. The guard cells are specialized cells which are responsible for regulating the entry and exit of water vapor, and also open and close to allow carbon dioxide to diffuse in to the photosynthesizing cells that need it, and also oxygen to dissolve out, to diffuse outwards. So here we can see the xylem and the phloem tissue within this vascular bundle, and here again we see the palisade mesophyll and the upper epidermis. So all of the tissues named in this plant section work towards one goal, maximizing the rate of photosynthesis. As the tissues work together to perform one or more definite functions, the leaf therefore is referred to as an organ. Now, I'm not going to fill in these gaps for you. If you're watching this video as a recording on my website, the video is about to pause and you can attempt this exercise for yourself. So see if you can fill in the gaps in this passage with the correct words from the bottom here. So vascular tissue, consisting of xylem and phloem in plants, is the main tissue responsible for transport of substances in flowering plants. A system of tubes called xylem vessels, which in the diagram here are coloured blue, are responsible for the transport of water and minerals upwards from the roots where they're being absorbed up the stem to the leaves where that water is used for photosynthesis. So in the xylem tissue, the direction of the transport of water is always upwards. Now the water and carbon dioxide from the air absorbed through the tiny holes called stomata in between the guard cells on the undersides of the leaves, these are all used in photosynthesis to produce glucose, a sugar, and oxygen. Now another system of tubes called phloem tissue, which is coloured red here in this diagram, is responsible for transporting the sugars made by photosynthesis from the leaves to the growing tips of the roots, but also to the growing tips of the shoots. So the phloem tissue actually transports sugars in both directions, upwards and downwards, through the stem of the plant to where they're needed. Now again, we're going to be looking at transport systems in plants in more detail in a later lesson of this topic. So here we can see the vascular system made up of the xylem and the phloem tissue. So you find this vasculature, this vascular system throughout the plant. You find it in the veins of the leaves, you find it in the stem, everywhere, because again, these substances need to be transported throughout the structure of this multicellular plant. The meristem tissue are the regions of tissue typically found in the tips of the shoots and also the tips of the roots. Plants will grow in height due to the activity of these zones of meristem tissue, which are found a few millimeters behind the shoot tips. So in the shoot tips, this region is referred to as apical meristem. Apical comes from the word apex, which means the tip of something. So if we take a vertical section through a shoot tip, here we can see the developing leaves, and in the bottom we can see the developing bud. And at this region here, the tip of the shoot, this is where, if we look under a microscope, we will find the meristem cells. These are a layer of rapidly dividing and differentiating cells, which are responsible for the rapid growth and differentiation of the tip of the shoot into these various structures, the developing leaves and the developing buds. Now, the roots at the other end of the plant also grow in length, and this is also due to the activity of zones of meristem tissue, which are again found a few millimeters behind the tips of the roots. Now, once again, this is also referred to as apical meristem because it's found behind the tips of the roots. So this is a diagram of a section taken through a root. Here we can see the phloem tissue. We can also see the xylem tissue here, but we can also see around the root, we see these root hairs which emerge from the epidermis of the root. In this region here, we have the meristem. This is the region where the cells are undergoing rapid division by mitosis and differentiation to form these various tissues as the root grows downwards into the soil. Now, to protect this very delicate region of meristem tissue, we have a root cap, which protects that region of cells as the root pushes downwards through the soil. And again, if we look at this region under a microscope, we can identify within the nuclei of these cells the stages of mitosis occurring here. Packing tissue is found in various parts of the plant. Good examples of this are the cortex and pith of the stem. The purpose of this is essentially to provide rigidity and also to divide the various types of tissue found in various parts of the plant. So here we can see a diagrammatic representation of this. In the center of this section of stem, we have the central pith, the packing tissue. And if we zoom in on this, we can see the cells. They're very nondescript cells. They're essentially there to pack this central region. So packing tissue can be compressed and will recoil upon release of pressure. And so this gives the stem of a plant the ability to bend in the wind without breaking. So once again, I'm not going to spend time filling this in for you. The video will pause and a question will pop up to allow you to attempt this activity for yourself. So have a read of this passage and see if you can drag the appropriate words to fill in the spaces in the passage for yourself. Now, the final type of plant tissue that we're going to look at is strengthening tissue. Strengthening tissue, which is composed of cells with thickened cell walls, is a good example of a specialized plant tissue, and this is typically found on the outer rim of the vascular bundles of xylem and phloem tissue. So here we can see a vascular bundle, and on the outer rim of that vascular bundle we can see these strengthened cells with thickened cell walls that make up the packing tissue. Here we can see the phloem, here we can see the cambium or meristem tissue, and here we can see the xylem tissue. Okay everyone, that's going to do it for this lesson. I hope that was useful to you. I'm now going to go into the Q&A discussion with the students that I have in attendance with me. Thanks once again for watching, and I'll see you in the next one. Take care.